and has served 32 years in CCS Haryana Agriculture University. And later, he has also worked in the Lala Rajput Rai University Veterinary Animal Sciences, HISA. He has retired as a Dean, College of Dairy Science and Technology on May 19, and he has contributed particularly the poultry disease diagnostic aspect and provided diagnostic services to the poultry farmers doing a vast postmortem examination around 6 lakh birds. It's a challenging. So he has awarded two lifetime achievement awards and contribution to the poultry sector. He has very good liaison with the government sector, state animal husbandry department, as well as the Indian poultry trade. He is truly a poultry academician as well as the poultry industry personnel. He is a bridge between both of them. He's taught UG and PG student, and he has guided to 7 MUSC and 3 PhD students. He has handled more than 10 research projects, including one with the University of Minnesota and as a principal investigator on the surveillance project on the Newcastle and avian influenza. He is a PI of another project of the University of Pennsylvania and PI of the ICR All India Coordinated Research Projects, one each on zoonosis and the veterinary epidemiology. He has published more than 87 research papers with international and national journals of repute, besides book, monograph, chapters in the book, and many popular articles in his name. He has visited 10 countries, including the USA, UK, Australia, China, for various scientific meetings and presentations of the research paper. At present, he is a member of Empowered Committee of the Animal Health of Department of Animal Husbandry and Dairying, Government of India for Policy in Foods. And also he is a member of Expert Working Group of Veterinary Products of Indian Pharmacopoeia Commission. And we have been pleasure to working with you on these various aspects. Presently, he is providing the consultancy to the poultry breeders and diagnostic facilities through Diagnostic Lab in Jind, Haryana. So I welcome you, sir, and request you to address the forum with the current challenges to the poultry industry. Thanks. Over to Vijay, please.
Yeah, thank you, Dr. Gaukare, for your welcome address. It's indeed a privilege and honor to have with us Dr. Mahajan, sir, a distinguished academician and also very closely connected with the poultry industry. So, sir, you are a kind of a bridge between academia and the poultry sector, and in your rich experience of over 3 decades, we are truly honored to have you, sir, and thank you so much once again. And now let me invite the inspiration behind the entire AAHP organization, our president, Dr. J.M. Kataria, sir, to please share us motivating words.
Thank you very much, Dr. Makhija. It is really a pleasure that again we are meeting here on the webinar. And for this webinar, I welcome you all. Dr. Mahajan, who is the speaker, having a very long inning into the active inning in the poultry industry, poultry disease diagnosis. Dr. Makhija, Dr. Gaukare, Dr. Sarvanan, and Dr. Reddy. I think Dr. Chatterjee has joined, or he will be joining subsequently. And all the participants who have joined this webinar or will be listening through the YouTube. It's really a great pleasure to have Dr. Mahajan today for this seminar, and he will be presenting on the current health challenges to the poultry industry. Of course, poultry industry development is definitely taking a very good lead. It is developing fast, whether broiler or layer production, we are going in a very good speed. But as you know, in between we are, Dr. Chatterjee is welcome, he has joined. Is definitely being put some hurdle in the development by the health aspects. After 2006, when the first AI was reported, it is an every year incidents will occur. Rather, it will be reported. We are not having only HPI, which is reported and culled and cured, but we are also having LPI, which has become now endemic, which Dr. Mahajan, I think he will be highlighting. And not only that AI is there, we have many other diseases like IB. IB has become so much endemic, and though various forms, various pathotypes, they are being reported in the country. And we have been using so many vaccines being used, and so many are still being imported. That is something very, causing a serious concern. We expect that Dr. Mahajan, who is actively associated with this type of free lancing service to the poultry industry, he will be able to put some point that how to make some impact on the control of IB. Similarly, last more than 10 years now, ILT has become a problem, particularly in the South region, which whether we have to use vaccine or not, it is a vaccine strain that is a big, it's still a complicated situation. We need to put a lot of efforts on these things. And in addition, we have IBH, we have MD virulent or very virulent, that is still we are arguing on it and not having any solution. We should, whatever we say as a scientist, but to industry, with the farmer, we have to give some solution. That what I have been talking, that every time we are going for culling and paying compensation, is it that the compensation works? How the industry will develop? Because the small farmer, they will be, scared to continue with the poultry industry. We will cull the farms. We have to come out with solutions. And another very important aspect, which I expect Dr. Mahajan will also deal, about the import of vaccine. Perhaps we have to be very, honest to our country's situation and not to import each and every strain, because DADF is bound. If they have allowed F from particular country, they have to allow the F strain from other country also. We have to see whether how much is the homology with our prevalent strain. And another last very important aspect is the diagnostic facilities in the country, which is perhaps, it is a very basic type of things, whether it is with the university, industry. Of course, there is the targeted, whatever the things they do, but in-depth studies are required. If strains are reported, we should go for their, epidemiology. Since Dr. Mahajan is a man of epidemiology also, he will be able to do make some clue about this. How to go for epidemiology is very important to identify which strain is the best for our country. With these words, I would like to thank the organizer for this opportunity, and I am lucky to have Dr. Mahajan, and I think it is Dr. Mahajan's turn to start the presentation. Thank you very much.
Thank you, Kataria sir, for overview of the current challenges with the poultry sector is facing. And indeed, we all of us are eager to learn and listen to Dr. Mahajan sir from his viewpoint, the current challenges and what could be the solutions for these challenges. Because you are the guiding light, sir, for all of us. With your expertise, we learn what kind of interventions, what kind of approaches, what kind of solutions you recommend to all the people on this call. And of course, not many people are watching this on the social media channel of ICR, DPR as well. So we are looking up to you, sir. And for the participant, this is the time we hear Dr. Mahajan sir. And in case you have any clarification, comment, feedback, question, please feel free to write in your chat box. And once Dr. Mahajan sir completes his presentation, we'll have the opportunity to discuss on those feedback. Thank you so much. And Dr. Mahajan sir, welcome once again.
Thank you, sir. So thanks, Dr. Kataria. for inviting me to this webinar series, which is a very good, we can think the association has taken up continuing with what we have been doing during COVID period, and this was a good one plus point from COVID, which we have learned. So thanks, Association of Avian Health Professionals, all members, Dr. Kataria, Dr. Gavkare, Dr. Reddy, Dr. Vijay Mukhija, and all my fellow veterinarian colleagues. So today I'll be presenting before you, sharing with you, rather not presenting, sharing my experiences, which I have a little bit during past more than 30 years now in Haryana. And of course, there have been a lot of ups and downs in the poultry industry, and we have seen it, and hopefully so coming to the poultry industry, Dr. Katari has already given the background, what we should discuss, and how we are having some issues related to health problems despite all advancements in the poultry production.
Sir, you can make it full screen, sir. Sorry to disturb.
Okay, it's okay. I just
make it full screen, sir.
Okay. Right. Is it okay now?
Yes, sir. Yes. Go ahead.
Thank you. Sorry for that. So we know that Indian poultry is the most organized and vibrant sector in animal agriculture, and having a value worth 1 lakh crores right now, and contributing more than 27% to agricultural GDP. So that is very good contribution compared to the other areas of agriculture. As per 20th livestock census, which came in 2019, our egg production has reached up to 103 billion from 46 billion eggs during the year 2005. And the broiler production has reached over 4 million tons. So these are good achievements, and maybe during this 4 or 5 year period, we must have gone much higher than this. So the growth rate of layers is 6 to 7%, while that of broilers is 8 to 10% per year, despite ups and downs which have been facing. But compared to agriculture, which is having only around 2.5%, these sectors are progressing at a very good speed. As per available data and the recommendation of National Institute of Nutrition, they say that at least one person should consume 180 eggs for a year and about 11 kg of meat. But we are lacking in both the areas in Indian context, that we are having only 79 eggs per person and 3.9 kg chicken meat respectively available. Compared to other countries, even in our adjoining countries of in Pakistan and Bangladesh, they have much higher availability of per capita availability of eggs in their countries. So India is still trailing at around 79 eggs per person per day, but it is maybe because our population too big. But we need to publicize egg is a vegetarian food, not a non-vegetarian, because there is still a stigma, and in the mind of Indian people, they consider it as non-vegetarian food. So government agencies as well as all NGOs or other agencies should try to publicize it, that this is not a non-vegetarian diet, this is a vegetarian food. And this sector is providing employment to over 3 million people. So that is a huge opportunity to people, and providing services to the nation and meeting the protein needs of the country. Coming to the Indian disease scenario, we have, particularly in the north, because most of the time I have been in the north. So here we find during April, when there is harvesting of wheat, it's the onset of very fine dust particles in the air because of harvesting. And at the same time, there is rise in temperature starts rising. And by May and June now, right now, May, it's very hot. It has gone to 43 degrees in Hisar, and at last night there was very heavy dust storm, so a lot of dust around us. So then now we can say many of the viruses which Dr. Kataria was saying, we have now these things they have become now endemic in our almost all poultry raising areas of the country. Baby H9, IV, NDB, some whatnot. Similarly, mycoplasma and in E. coli coming to the bacteria, they are also there because mycoplasma is coming vertically also, and E. coli is wherever you grow chicken, E. coli will be there. So then we have been facing emerging flus like bird flu in H9 and or H9N2. Maybe as we know that this H9N2 started way back in 2003 when it came from Pakistan into our country, and when H5N1 it came in 2006, and then we kept on we kept on experiencing outbreaks of H5N1 somewhere, some part of the country, wherever migratory birds used to visit. And during 2016, we found H5N8, and I was actually involved in this outbreak, one which had happened at Hisar also, because it was a tourist complex where there is a lake and there were a lot of ducks they were raising there, and this disease came in those ducks. So I had performed the postmortem also at that time, first postmortem, and then said that, well, we should not open these carcasses and we should straightway send the sample to Bhopal. So immediately we sent the sample and it was then confirmed as H5N8 in 2016. And then 2020, again, we had a very huge outbreak. Almost, I started from again in Barwala area in Haryana and went up to spread all over India. So then there has been change in the poultry industry as such because of whatever we had been earlier seeing when I started in my career in poultry, maybe way back in 1986. There used to be very small farms like 2,000, 3,000, 5,000, and 10,000 farm used to be very big. I mean, he's a big farmer having 10,000 birds. But now you see 10,000 is not counted at all. They say, oh, small farmers, 10,000. And most of the farms, they have become lakhs of birds at one place. So the things are changing. And now earlier we used to sell, market the bird before maybe 1.5 kg up to 2 kg. But now people are preferring it to 3 kg or even beyond. And for the past about 3-4 years, there has been a lot of companies coming into the integration sector, and a lot of particularly broiler farming has gone almost 90% into integration companies, and hardly 10% people, they are doing their farming independently. Then there has been a lot of thanks to Gadkari ji, because he has built so very good roads all around the country, and because of that, movement of eggs, chicks, and far off places has become very common. And we may, because of that, the chances of transfer of infection from one place to another is also there. The less rigid biosecurity practices, particularly in this part, what we can see is that the lifting vehicles which come to collect the birds, they're not doing the biosecurity practices so seriously. Though the farmers themselves should enforce them, that they should not load the bird before they bring the vehicle properly disinfected. Unfavorable environmental conditions, of course, right now, as we are having right now in north, temperature soaring to 43, 44 degrees Celsius right now. And birds who are in open houses, they are having open mouth breathing. Of course, now many farms which are bigger farms, they have gone to EC houses, so that is a plus point. So there have been in the field situation, but I could see particularly I am dealing with poultry broilers as well as breeders. So immunosuppressive viruses, they are very common. They have become very common, particularly in the broilers like IBD and chicken anemia virus. Then in the past 6 months, I'll come to that, what problem we had in particularly in the broiler sector. And the mycotoxins was one of the major reasons. Respiratory viruses, as we all know, that they have become all endemic in here, and Mycoplasma E. coli, and then climatic factors. Right now, just like right, we have high temperature and dust storms. So the combination of environment, virus, Mycoplasma, and E. coli put together, it's giving rise to respiratory disease complex, which is very difficult to handle and causing lot of mortality, particularly if young chicks in broiler sector. So what are the reasons for immunosuppression in poultry? So there are many reasons. Of course, we all know that, we all being veterinarian, we all have been knowing it. So I'm not giving much of the lecture in those directions, but I'm just trying to emphasize that these are the points which are very important to be considered at the time of disease diagnosis and for advising the farmers what to do. So immune system of birds, we will discuss that. It's little different than what other mammals or other warm-blooded animals they have. So infectious causes, they are like various viral infections which can lead to immunosuppression. IBD is the number one, followed by chicken anemia virus, MD, IBH, Rio, and any others. Then coming to non-infectious causes, mycotoxins is one important area, and this being the major component of feed, being the major component which is required for raising the poultry. So it's very important that we should consider it also into our diagnosis. Stress, poor managemental practices, they can also lead to immunosuppression. But it's mainly a combination of all these factors put together, and they give an additive effect or aggregative effect, which for now you can say they amplify the immunosuppression further. But there are many times there is subclinical immunosuppression also, which is an important but often underestimated factor, which is important for subsequent development of clinical disease. So this is avian immune system. So we normally know that the Bursa fabricius, which everybody who's dealing with poultry, even farmers, they know that Bursa mein kuch gadbad hai. People, they say that possibly there is IBD, just there is enlargement in Bursa, or there is atrophy of Bursa. Then thymus, we can see now in the early stage of chick, particularly 2 to 3 weeks age chicks, the signs of chicken anemia virus, where there is atrophy of thymus gland as well as in bone marrow. Then gut-associated lymphoid tissues, fecal tonsils, spleen, and Herderian gland. These are some of the organs which play important role in producing the immune response and in their immunity in birds. So why viruses are a major problem to control? Because there is plurality of serotypes. Not many serotypes are there in one particular virus. Many fill strain and many pathotypes. Then persistence, once disease comes into a farm, particularly I'm just giving one example of IBD. If IBD comes in a farm, so it is going to stay there if the biosecurity system or his disinfection is not proper, because the virus can survive up to 4 months at the same location, through in the litter or somewhere on the walls or something. So because of that, it's very difficult to control such type of these viral diseases. Then there is evading of immune mechanism, and there is change in antigenicity as the viruses they come under pressure from antibodies. They change themselves, and new variants they emerge. They are independent. They can independently survive in the host, and there is very fast spreading disease. Most of this we can find out from the farmer also, that what was they come normally with the complaint that yesterday he could find only 4 cases, and today they are more than 100. So and the speed next day it will be more than 1,000. So it spreads very fast, and lesser susceptibility to common disinfectants if they are not aware about the farmers that how much dose they should use, which disinfectant they should use for that for different viruses. The transmission through insect vectors is also possible. So that is another area where we have to look into, and because they are beyond our, many times it's beyond our control. And many of these viruses, as we know, that they cause immunosuppression. So economic losses due to viruses, they cause very high mortality. There is no medication for viral diseases. So only and the secondary infections are definitely they will take over subsequently as the viruses they go away. And the secondary infection, they require a lot of tons of antibiotic to be pumped into the farm, to the birds, and that adds to our AMR. Again, because it then lot adds to our cost of production as well as problem of AMR. So and then these viruses, they cause loss of production, deterioration in quality of products, and in some certain diseases like AI, there is trade restrictions for export to other countries. And so how the emergence of viral diseases or syndromes, they are happening. So now we know that there is intensification of poultry rearing system all over the country, and big farms are coming up. So there are commercial regions also, because trade of birds, products and vaccines across the states and across the countries. And Dr. Kataria rightly put that we are having a lot of vaccine from abroad and a lot of strains, particularly for the live vaccine, we have to be very careful. Definitely, sir, in the committee, we are trying to see this thing that we do not allow unnecessarily the strains which are not homologous to our circulating viruses. So multi-age farming, this again adds to the problem of that emergence of new viral disease, viral syndrome. Then we have free living birds around the farms around us, aquatic birds, migratory birds. They may carry the viruses, migratory birds particularly, they carry the viruses from different parts of the world. They come to our place, our country, particularly during winters. Mutation, recombination of live vaccine and field viruses. This is another area where we have to look into whether any new recombinants they are coming because of use of excessive live vaccine along with field virus. Genetic makeup of birds, because now as we know that the genetic makeup of birds has been increased. There has been a lot of improvement in genetic makeup of the bird. But when you improve productivity of birds, definitely you are losing on immune aspect, because these two things may be negatively correlated. So there are reservoirs of some diseases in nature as well as in the wildlife, wild birds. So coming to infectious bursal disease, which is a major immunosuppressive virus, and of course I will not go in much in detail, but I can say what we have visualized in the past about 5-6 months is that earlier we used to say that IBD comes around 3 weeks, after 3 weeks, 3 to 6 weeks time. But now we are seeing lesion before 13 days of age, when normally we used to vaccinate at 13 days of age. And even the bursa in a 10-day-old broiler will be completely atrophied. Atrophied, or it will be having some exudate. So agent, it is the different types of classical virus variant and VV-IBD, causing heavy mortality, and as a result, a lot of immunosuppression, because the immature B cells and bursa fabricius and lymphoid cells, they are the targets for this virus, and they destroy those. And as a result, birds are not able to protect themselves from the challenges, and there is failure of vaccination because of these things. So this type of clear-cut lesions may not be so common, but we are seeing that the age of infection, age of birds showing clinical lesion has come down. And coming to prevention of and control, what we normally used to do is the most adopted approach was to hyperimmunize of hens. So we used to give 2 live vaccines and 2 killed vaccines so that they can pass on the maternal antibodies to their chicks for at least protection for a few days after the chicks hatch, to maybe 7 to 10 days. Then there was since the 80s, we were having a Cohen-Hoven formula for collection for calculation of day of vaccination chicks, but that was actually not being followed in all the hatcheries because of so many variation in vaccination, and depending upon the market value or market of price of chicks. If it is high, they may vaccinate the parent with all the vaccines, but if it price goes down, they may skip some of the vaccines. So as a result, the maternal antibody status in day-old chicks was very much variable. And I had given this problem to one of my student who was doing masters, and right now she's handling my laboratory at Gene, where and she had worked on for this, and in different hatcheries of Haryana, we had tested the maternal antibody status, and it was very variable, starting up 6 days to 16 days. So it's difficult to say at what is the right. So it varies for each hatchery. Live vaccine and 2 or more inactivated vaccine in replacement pullets, and hence that is common practice, which is being followed, has been being followed. And progeny is protected by passively derived and maternal antibody that we know. And live vaccines are now being used in ovo or at hatching also. But recently there has been recombinant vector vaccines. They have come, maybe I have seen them for past 4 or 5 months in this area, and people are using, particularly the integrators, they are using it on day 1 or getting it vaccinated in the hatcheries itself. And we could find that it is giving a broader protection against most pathotypes, and it is a single vaccine given on day 1 or at the time of hatch, lifelong immunity in broilers against IBD with no immunity gap. So next one is chicken anemia. Of course, we know that it causes plastic anemia in commercially produced chickens, and is prevalent in laying and broiler chickens. I could see that incidence in broiler chicks is gradually increasing. It was not there for, during last 5-6 years, but now I could see that in many hatcheries it's coming up. How and from where it is coming, that's the question. Because the disease is transmitted vertically, so we have to see that, and accordingly, if subclinical infections are also common, so likewise this can cause this type of problem. Secondary infections, mainly in layers, it's very common, leading to Clostridium perfringens, Staph aureus, and leading to gangrenous dermatitis. But it's not so common in broilers. But definitely, immunosuppression is there and anemia is there. So it causes because of immunosuppression. If there is a concurrent viruses like IBD, then it is going to be very tough to. The bird will be too much immunosuppressed if both of them are present at the same time. All ages are normally susceptible, but usually we find between 2 to 4 weeks of age, chicks are more susceptible, and they are showing typical lesions in the bone marrow as well as in the thymus and in the anemia. So a CAB infection is subclinical in older bird normally. So there is a vaccine available, so we can try this vaccine also, particularly in the commercial layers or breeder birds. Then coming to the feed, which is one of the major input, we can say more than 70% of expenses on poultry farming is on feed. So it is very important for optimum production, whether egg production or body weight or FCR. It provides all nutrients which are necessary for growth, production, and immunity. However, because many times it depends on because we use a lot of maize which is a major component in all type of poultry in our country. So mycotoxins, which are now invariably found in different concentration amounts depending upon from what is the source, from where they are importing or they are getting the maize, which season. Then we know that many of these mycotoxins, they lead to immunosuppression also. So major ingredient in feeds, as we know, is maize, cereal, bajra, groundnut cake, sunflower cake, soybean meal, rice polish, etc. So as per FAO data, 25% of world's grain is contaminated with mycotoxins annually. And we know that these crops are harvested seasonally, and they have variable moisture content, and even within a lot, because maybe small grower, there are small growing farmers, they may be selling few sacks, some other big farmers, and all put together at it by the distributor. And then in some of the bags, if the moisture is more than 13%, fungi will start growing, because fungi normally contaminate even at the time of when they are growing in the field. So these grains are required to be stored for longer time, because they are grown once in a year and transported long distances. They are coming from, we are getting mainly maize from Bihar and UP and Rajasthan, and even from south in Haryana. So fungi, we know that they are ubiquitous and they contaminate growing crops and grains. And during storage, they grow and produce toxins at being a very high temperature, more than 30 degrees Celsius, and the relative humidity is more than 80 to 85%. And if moisture content is above 13%, so it's a very good environment for growing production of toxins in that situation. And particularly the coming season, when in July, August, we will have all these conditions, very conducive. So if the crop is having already contaminated with fungus, so it is going to be a problem. So during rainy season, and this type of things could be there. So there are thousands of chemically distinct mycotoxins. Most depend on the chemical structure, and they also impair protein synthesis. They exert different effects depending upon the level of contamination and duration of ingestion, how long you are feeding same product for longer, when it definitely causes damage to the liver and kidneys primarily. And this was very a peculiar feature all during past about 6 months in broiler farming, whether any feed or most of the feeds. Overt clinical disease, sometimes when it's very high mycotoxin content. And there was, but recently, in about 15 days earlier, there was a mortality of around 18,000 birds in 3-4 days, because of he had used a very old feed. Just he said that now my birds are getting ready to the market, and let me finish this feed which was lying there for quite some time, for 2-3 months. And he just fed them for 4-5 days, and within that period of time, all birds, almost hardly 22,000 birds were saved. Rest 18,000 died. So he had to suffer a great loss. Reduced feed intake. It reduces feed intake and weight gain, impairs immune system, makes the bird more prone to particularly IBH, was very common, followed by, if initially it is having mycotoxin, so liver is already under stress, and if IBH, which may be latent, otherwise not causing that much problem, it becomes active and starts multiplying and produces more problem, and mortalities increase, even leading to sometime hydropericardium. In breeder layers, they reduce reproductive efficiency and hatchability. So there are a number of mycotoxins like fusarium. Aflatoxin is number one, which we are normally experiencing and also testing them. Most commonly, we are testing for aflatoxins, but not going for a test for all the toxin all the time. So we can say that aflatoxin is a indicator toxin. If we find aflatoxin there in the feed, it is quite possible that the feed may be containing other toxins as well. So one or many toxins may be found together, and they act synergistically. And we normally test for level of aflatoxin mainly. And in such situation, mortality can go as high as 1 to 40%. So these toxins, they are normally resistant to digestive processes and they get absorbed. So these are, you can say, the maize is one substrate on which all different types of fungi they can grow and produce toxins. They can be hepatotoxic or nephrotoxic. Most of them are hepatotoxic and nephrotoxic. So this is how we can see that, on which part or which organ these different toxins they act and affect the bird. There could be swollen kidneys, enlarged liver, swollen kidneys, and bending of sternum. These are some of the PM changes which we can see. So it is always, prevention is better than cure, we always say. So it is always better to prevent the formation of mycotoxins. It is more economical than treat after the birds have come down. So we should guide the farmers regarding proper purchase and storage of grains and feed. Because many farmers, when we ask, they do not know where to store the feed. They keep it on the floor itself without. It may get moisture from the surface floor itself, and then temperature inside the shed is high, so it gives a very conducive condition for production of mycotoxin further. Once produced, these mycotoxins, once they are produced, they cannot be inactivated by feed manufacturing process or by storage. Chemical inactivation has been tried, but they have not been found practically feasible. But many toxin binding products are there now, which help by absorption, like zeolites, and other combinations are there now within the market, which are very good and they are giving better results. So we should go for those regular use of those good quality toxin binders in the feed. And testing of maize, particularly before unloading of the vehicle from the vehicle, we should carry out from different sacks a very comprehensive sampling and then test it before unloading, so that we can find out what type of grains we are having. And regular use of organic acids, they are also effective, good. They help in not allowing the growth of mycotoxins, propionic acid, formic acid, benzoic or acetic acids. And they are also help in prevention of insect infestation in the feed or in the crop or in the grains. Enzyme produced by yeast bacteria, they are also useful to take care of mycotoxic effects in the birds. Similarly, sulfur-containing amino acids, antioxidants, and the change in feed and grain. If you find that there is toxicity in the birds showing in postmortem, immediately ask the farmer to change the feed. May use some grain for 24 hours and then subsequently use a fresh source of particularly the grain which you think is free from mycotoxin and having moisture content less than 12%. What is the permissible levels and mycotoxin? That is again very subjective. Normally we say that 20 parts per billion of aflatoxin, and beyond that it is toxic. So keeps on varying, but it depends upon whether it is a aflatoxin alone or if there are combination of other toxins also. Because when you find that despite giving liver tonics, you are not able to correct the birds. So you correct their health. So you have to look for other toxins also sometimes, get them analyzed. So this was the field problem in broiler during past 6 months, which we have faced in North. Combination of factors. One was source of chicks or day-old chick quality, because again, as I told you, the market during the past 1.5 years, even after COVID, particularly for breeders, has not been very good. And they have seen a lot of losses, and because of that, obviously they were forced not to go for skip some of the vaccines and some of the feed supplements in the feed. So problem may start in 10 days to 2 weeks age broilers, and liver, kidneys, they are extensively enlarged, showing some hemorrhages, mild exudate in bursa, or even atrophied bursa, as well as anemia. Mild HPS, IBH, and diarrhea. Livers are pale, fatty, and enlarged. Kidneys swollen, hemorrhages, and urates. The role of meat and bone meal, well, that is again questionable. We cannot find because there are so many different sources, whether they are ISI mark or not. We are not sure about that. So when you give, and there were people were tried different liver tonics, but they were not effective. So mortality was variable, 7 to 20%. Now also with the after the April month, there is LPI, IB, mycoplasma, and E. coli, and the mortality has gone up to 50%, particularly in the young growing broilers with up to the age of 3 weeks time. Mortality is too big because of in bronchi, they are completely clogged because of mucus or because of exudates. So they are not able to, they're gasping and then they are dying fast. So immunosuppression is a major problem. So appropriate vaccination, biosecurity, along with mycotoxin management is very essential for broiler industry. And coming to the LPI, LPI is universally present in poultry and with varying degree of pathogenicity, and prominent among these are H5, H7, or H9, but we are having H9 particularly in our region and in our Asian region. So respiratory signs, sneezing, ocular, nasal discharge, swollen infrared sinuses, open mouth breathing, this is very common. And drop in egg production in layers and breeders, misshapen eggs, decreased fertility, hatchability, lethargy, particularly in broilers, there is a lethargy, they stop feeding, they stop taking water consumption, and it spreads very fast and increased sprout. There is very fast increase in morbidity and mortality. So postmortem lesions, congestion and inflammation in the trachea, the secondary bacterial infections, required caseous exudate, edema, pneumonia, and air sequelitis is very common. So this has become very common, air sequelitis, recently. Lesions, hemorrhagic ovary, involuted and degenerated ovine layers, and yolk in the abdominal cavity and peritonitis in laying birds. Then hemorrhages on serosal surface of proventriculus and gizzard. This is very common, and this is very, you can say pathognomonic also sometimes. Enteritis, a small number of birds may have signs of acute renal failure and even visceral urate deposition. So now you can see the tracheal mucosa show hyperemia, petechial hemorrhages, and caseous exudate. This type of things, even air sequelitis and perihepatitis, pericarditis, and yellowish discoloration, this is very common. So indicating some mycoplasma as well as E. coli combination. This is the hemorrhages on serosal surface of gizzard. And this is now you can see in laying birds, this peritonitis. So the severity of disease depends upon the virulence of the virus, immune status of the bird, accompanying concurrent infection or secondary infection, and what are the different stressors on the bird. As right now we have a high temperature and dusty environment, which is helping them. What are the routes of transmission? Like fecal oral route, virus survives for 7 days at 20 degrees Celsius, and in liquid litter, like in layers, it survives for 105 days. Sick or recovered birds, so we should go for grading, and immediately once we have start this problem in the flock, we should ask farmer to grade the birds and remove the affected bird as early as possible. Start spraying in the shed disinfectants which are effective, like various Pircon or other different B904. There are 9 number of products available, whatever is effective as per their experience. No vertical transmission. Virus can be present on the eggs of laying birds because of contamination of feces through feces. Waterfowl, migratory birds are the natural reservoirs of these viruses also, and they transfer the infection. Improper disposal Improper disposal of infected carcass, manure, or poultry products is one of the reasons also for vast spread of this problem. As farmers, they are not aware that how to dispose of the carcass or as well as manure. And transportation or sale of affected birds across states or markets. And we had seen during this H5N8 outbreak from Barwala area, a lot of these layer birds where they were sold all over the country at cheaper prices, and the people suffered in those areas who bought those birds. People and equipments, through people and equipment, it can be through fomites, it can be transferred, insects and rodents. So what are various physical and chemical actions which these viruses can resist? Like it can be inactivated by high acidic or high alkaline pH. Either pH should be less than 3 or it should be more than 10. So very alkaline solution or very acidic solution will kill virus, including H5 or H9. And it remains viable for long period in tissues, feces, and also in water. So inactivated by oxidizing agents, sodium dodecyl sulfate, lipid solvents, beta propiolactone, inactivated by formalin and iodine compounds also, but that depends if there is no organic material along. Then diagnostic tests. These avian influenza viruses can be detected in oropharyngeal, tracheal, or cloacal swabs from live birds. And from dead birds, we can collect samples from internal organ like trachea, lungs, and air sacs. And they by RT-PCR and sequence analysis of the viral HA or NA genes can confirm the virus. And genetic tests can be carried out to identify the characteristic pattern in the HA at its cleavage site, and virulence tests in young chickens to distinguish it whether it is LPI or HPI virus. So they are done maybe in Bhopal lab mainly. So incidence of H9N2 LPI in Asia, it has been reported from various Asian countries all around us, like China, Israel, Egypt, Iran, Korea, Pakistan, Bangladesh, India. So H9N2 viruses have been reported in countries sharing our porous border with us, like Nepal, Bhutan, Pakistan, and Bangladesh. They have reported very high incidences of H9N2 LPI, which have caused losses in those countries, and both countries showed close relation to G1 lineage. Few cases of human H9N2 were also reported from Bangladesh in 2011 and 2015. Many researchers in India have also now isolated H9N2 viruses from different farms across India. And among the Eurasian virus, H9N2 of G1 lineage have been the most prevalent in South Asia. And we know that LPI is an overt loss-causing clinical disease due to H9N2 surfaced in 2003 in India. So, as I told you that this came from Pakistan in December 2002 in Tarantan area, which is adjoining in Punjab, very adjoining to Pakistan. And we can also remember that we got the Ankara disease or the HPS also through Pakistan long years back. So this disease came again from there, and then it came to Pathankot in January 2003. From Jirpur it reached Barwala, and 10th of March it was confirmed in Barwala area, and since then now it has spread all over India. So you can see, so it is a very devastating disease, and a lot of losses it has caused to our poultry industry. So the control strategies, there's no treatments. We have to go only for vaccination and biosecurity. Why vaccination is necessary? So this was the question which we were putting before our government agencies, that we should allow vaccination, because disease has become endemic now in India, and so there's no other alternative to control this viral disease. So endemic infection with heavy economic losses. And since for the past 20 years, we have been seeing different, almost everywhere, this type of problem across Indian subcontinent, including Pakistan, Bangladesh. And as I told in the initial, we require a lot of antibiotics to control secondary bacterial infection because of this infection. H9N2 may itself evolve into pandemic influenza agent, which is again a question mark agent by reassortment or co-circulation with other subtypes in migratory birds, leading to newer mutants in the domestic poultry. So this is again a risk which is there. These two authorities on avian influenza, Capua and Swan, they have said that strategic use of vaccine in conjunction with other control measures can be effective in controlling AI virus. By vaccination, what we are doing is it is decrease the environmental contamination is decreased by decreased duration of excretion of virus and decreased quantity of the virus. This is true for both avian influenza as well as H9. So vaccination reduces the total amount of virus contaminating the environment. So whether it is HPI, LPI vaccines, AI vaccines are produced using various technologies, including inactivated whole AI vaccines. As right now, our government has allowed us in vitro expression HA protein, in vivo express HA protein in vectored system, and HA-based DNA vaccine to induce protective immunity. So, but question is that we have to, AI vaccine must be periodically re-evaluated to determine whether they are still effective against the circulating field virus, because the viruses keep on evolving very fast. So we have to be updated. Similarly, we have to update our vaccine strains. Only then we can be able to protect our poultry. The current situation and concern that H9N2 has surfaced as an economically threatening disease during the last 20 years, with increasing number of countries having to resort to vaccination for prevention of LPI alongside HPI infections. So recurring outbreaks of H5N1 year after year since 2006, and now with H5N8 and 2016 AK21 must acknowledge the fact that H9N2 and H5N1 co-circulate and new assortments can emerge, resulting in undesirable impact on poultry and human health. So that is another a risk area where this can lead us to. So in the sense of policy of vaccination against HPI, it becomes all the more very important to at least reduce and eliminate H9N2 virus from the scene. So this is what we try to emphasize to government agencies, and Dr. Vijay Makija, Dr. Gavkare, we all were part of the committee which we had put forth our point along with associations of Poultry Federation of India and other bodies to the government. Having established that H9N2 LPI is a major hurdle in sustaining the growth of poultry industry, there was urgent need to acknowledge that avian influenza in subcontinent, and particularly to convince the Ministry of Fishery, Animal Husbandry and Dairying was difficult, but we could finally convince them for legalized vaccine production. Now, NISAD Bhopal will monitor and study in detail the molecular characteristics of prevalent H9N2 viruses across country and keep updating on emergence of new variants. And NISAD has selected an appropriate candidate vaccine strain based on molecular cartography, serological and animal challenge methods, and given this technology to Indian vaccine manufacturers. And then they will have to keep updating every 2 years on usefulness of existing vaccine strain, along with seromonitoring, etc. So the Indian vaccine manufacturers, to produce a properly regulated, high quality region, country, and clade specific vaccine now, they can produce it for Indian consumption as well as maybe for export also. Poultry industry to send appropriate samples to RDDLs or NISAD Bhopal for surveillance and updating of vaccine strain, and for monitoring efficacy of vaccines and for surveillance purpose also, because that is required. We need to keep on sending samples to RDDLs or NISAD as agreed by government, so that we keep a watch whether the new variants are emerging. However, we should emphasize more on strict biosecurity practices, which should become a permanent feature of all poultry farms operations to minimize the risk of various viral infections. Then coming to NDV, which again we have been facing some new variants in NDV, which 10 years earlier we were not so much worried about that, but during the past 10 years there have been a lot of new genotypes emerging. Like there were 18 genotypes in class 2 and only one genotype in class 1. So total 19 genotypes, but recently there has been one more genotype which seems there, so which caused a lot of mortality in some areas in Haryana also. So we know that these antiviruses, they are viscerotropic, neurotropic, mesogenic, clantogenic, and asymptomatic. But most pathogenic ones, they are normally velogenic and mesogenic in between. So pathogenic viruses, they will have this type of fusion gene cleavage site compared to the low virulent NDB virus. So again, we know that this is endemic in almost all, though we do not have a very systematic data available with us regarding surveillance, but we can say that on the basis of outbreaks and in intensive poultry farming areas, everywhere we find we find the Newcastle disease incidence. Shedding of virus remains a very big challenge. And emergence of new genotypes in Haryana, we have noticed high mortality in peacocks in 2012, and I was one of the I was personally associated with this outbreak investigation, and genotype 7 was isolated. And because normally we consider peacock is a very hardy bird, and it is a wild bird, and normally survive most of the natural small challenges, but it started dying, and then we could isolate the virus from these peacocks. It was confirmed to genotype 7. Similarly, in 2018, again, there was mortality in peacocks, which was noticed, and the genotype 13 could be confirmed. And now a new variant, genotype 7I or genotype 19, in some parts has caused heavy mortality in layers and breeders despite routine vaccination, which they are giving. But now we have all the vaccines, kill vaccines, they have to be updated as per existing these new genotypes. So this was the publication which we got from this paper during that time. It was in peacock. So the class 1 isolates mostly they are grouped into genotype, whereas class 2 isolates are divided into 1 to 18 genotypes. Most of them are considered to be virulent except 1, 2, and 10. So clinical diagnosis based on history, signs, and lesions are fine, but we must get the laboratory confirmation to find out which genotype it is. The next generation sequencing is one of the most recent tools that have revolutionized the diagnosis of infectious diseases. It facilitates the rapid, sensitive, and specific detection and differentiation of mixed infection within a single host. So we can find out the respiratory virome and find out what different types of agents are present in that particular tissue or host or in the bird. So currently it is the most thorough tool which is used in discovery of novel viruses associated with unknown diseases, and like minion sort of one small equipment is also available, sort of which can be used in field level work to carry out smaller studies in field situation. Most promising vaccines against infection in poultry could be recombinant or vector vaccines generated by reverse genetics, having wide coverage and no risk of virus evolution. I had one outbreak in breeder flock also way back in 2019, and there was, despite vaccination, there was a lot of mortality going on. And it was a flock of 60,000 birds and 7 weeks of age, showing typical lesion in proventriculus as well as torticollis and the brain. And brain was also affected. So mortality was 12,500 birds, and genotype 13 was isolated. And I could, because of epidemiology, we could trace the source of infection, that it was from a laborer who was having desi birds in his home, and they were also dying. From there, it came to the farm. Right. So we have to be careful. We have to ask the history from the owners, from the laborers, so we can reach at it and what could be the source of infection. So recently, we have published one complete genome sequence of Newcastle disease virus isolated from backyard chicken in northern India. That this was part of that US project with University of Minnesota, which we were doing surveillance, and we could find out the two strains and one genotype 13C class 2 isolated from genome sizes. These two genome sizes were identified. So there are different types of vaccines which are available for Newcastle disease, like F strain, lantogenic strain, etc. So, but we have now we have to go for either for in vivo vaccination antigenically matched vaccine. But now there are various recombinant vaccines like available smallpox vectored, herpes virus or turkey vectored, or recombinant NDV vectored vaccines. So as I told you that we have to update our Lasota, this kill vaccine also with the latest genotypes. So a real challenge in NDV is detection of all NDVs, all genotypes of NDVs. The role of vaccination in virus evolution, this is again a question mark, whether live vaccines are helping in evolution of new genotypes. Vaccination may give protection from disease, but may not stop virus shedding. So, and large phylogenetic and antigenic distance between vaccine, this provides a scope for virus evolution. And there could be rolling reactions due to suboptimal vaccination. Low virulent NDV need only 2-point mutation to become pathogenic. Similarly, other issues like spillover, host passage, expanded host range may lead to virus evolution. So then, as Dr. Kataria was saying, that in infectious bronchitis also, it's again a very huge topic. So I have not gone to that detail, but I'm just touching it. So infectious bronchitis is again a very huge problem, and it has a lot of serotypes. And we know it's a coronavirus; it keeps mutating very fast, and several serotypes and strains of IV exist. And respiratory diseases are very common, particularly in the young growing chicks. Nephritis, enteropathogenicity, myopathy, drop in egg production, poor quality eggs. These are all there. So this type of problem in broilers were at least 2-3 weeks of age was very common. This is in laying birds, this type of shell problems and drop in production. So what we get on postmortem was that serous catarrhal caseous exudate in trachea, nasal cavity and sinuses, caseous plugs of mucus in the primary bronchi, tracheal plugs at the bifurcation of trachea into the bronchi. So it was clogged and bird was not able to take respiration and die. Congested blood vessel in the trachea. The lungs may appear congested and air sacculitis is also common in this case. So nephropathogenic strains, sometimes they are also there. So we have to go for vaccination again, but again, it's a big question, which type of vaccine we should go again. We need to find out through, obviously, we have to carry out surveillance for in different areas, particularly in the poultry raising areas, to find out which type of virus is prevalent in that area, so that our vaccination is properly directed and it is effective. And there are certain vaccination reactions, and in particularly in broilers, that if you give a very hotter vaccine or can say less attenuated vaccine, so it can lead to problems in the broilers. To prevent viral diseases, use of vaccine only way in endemic areas, we know that, but with live respiratory viral vaccines, which can replicate in the bird, they cause some degree of cell damage. If the birds are healthy, in a good environment, there will be minimum reaction. But if the environment is not conducive, or it's worse when the temperature is very high, severe vaccination reaction following ND, IB vaccines, plus E. coli infection have been the cause of respiratory complications. Similarly, immunosuppressed birds vaccinated with live NDV, they have led to severe vaccination reaction. Particularly, we vaccinate birds during the first week of broiler with the Lasota vaccine or any other hotter vaccine. So spray vaccine, similarly, very fine sprays are produced, and access of these vaccines to deep respiratory tissues can lead to excessive replication of CRD and RDC. So maybe in our situation, we have to standardize this spray vaccination. So far, it is not properly standardized, particularly in North. People have tried, but they have not succeeded in getting it good results out of by spray vaccination. Similarly, most of the farmers, they go for drinking water vaccination, which we know that it is not a right way of vaccinating. Intraocular or intranasal is the best. First vaccination should always be given through intraocular intranasal route. But because people require a lot of labor with big farms, so they avoid it and they just give it through drinking water. But that's the uneven vaccination. So respiratory viral vaccines of various virulence levels which are available, they can be chosen according to the proper specific condition, areas, type of bird, age of bird, etc. only then we can control these type of problems. Similarly, last attenuated vaccine like Lasota MA5 of IB, maybe if given in older bird, it's fine, but if given in first week of Borrelia, it will be difficult for the bird to handle. So recombinant vaccine for these three combinations sometimes may be effective, may be good, particularly in Borrelia. We have seen one vaccine which is having, it is a Merck's HBT vaccine which is having Newcastle as well as IBD recombinants. So that means single shot you can protect birds from all these three diseases. So good managemental practices have no alternatives. We have to always adhere to good managemental practices because they are the one which lead ultimately to towards the disease development. Because if the management is poor, that is predisposing bird to disease. So we have to be very careful in particularly during summer in housing management, providing maintaining temperature inside the farm to up to 22 to 25 degrees. Water management, because consumption of water increases as the rise in temperature increases. So we have to provide a good quality water. We have to test the water regularly for life forms as well as for chemical composition, because poultry have the tendency capacity not to tolerate more than 0.5% salt through all sources, maybe water or through feed. So those feed fortification and feeding management, particularly during summer, is very essential. What should be the timing of feeding? General management, all these put together, they are there. So there has been a lot of talk that people have been claiming that veterinary sector is contributing a great deal in antimicrobial resistance. These were some of the areas where through which they say they are contributing towards the AMR in human population. So how we can help in reducing it or this challenge? Even I have faced myself that many of the antibiotics which we are using in bulk, because most of the farmers, once they start using one antibiotic, they keep on using it regularly for maybe 5-10 batches continuously, and then one day they will say that it's not working. They make the dosage double, they make the dose triple, and then still they say that it's not responding. So this is how we need to educate them that there is correct, accurate identification of the causal microorganism is essential. Find out antimicrobial sensitivity pattern, just to find out appropriate drug of choice, not to waste of money on unnecessarily pumping antibiotics to the birds. So conclusions: viral infections like NDV, IB, H9, H5, and their evolution into newer genotypes or strains will remain a constant threat to poultry industry, and we all need to keep a watch on that. Need for judicious use of proper vaccines for different types of birds in different geographical areas, depending upon prevalence of or type of virus in those areas. Regular upgradation of inactivated vaccines and diagnostics as per existing field virus, that is a must. We have to keep on updating our field vaccines also. Regular monitoring of titers by ELISA for particularly ND, IB, at least these for we can monitor, should give us some idea how our flocks are performing. Then confirmation of virus, because we always say whether any farmer or any veterinarian, they will say there is viral problem. There is viral problem. Which virus? What type of virus it is? We need to get it confirmed. So for confirmation of virus, we have to go take a help of a diagnostic lab, go to our PCR, real-time PCR or qPCR to reach at a conclusion what is the culprit, which is the culprit, and why we are having that much mortality. Follow ABST to control secondary bacterial infection in wake of AMR, but just to get good results in treatment also. And last but not the least, robust biosecurity and managemental practices should be strictly followed, because they will always be required in the prevailing situation when we have a lot of challenges from viruses and lot of challenging bacteria and environment. So thank you very much for all for listening patiently. But I have not done much much in science, but whatever little I knew from my experiences, I have shared. Thank you so much.
Thank you, Dr. Mahajan, sir. Indeed, it was a very comprehensive overview of the wide variety of challenges the poultry health sector is facing. And you covered right from, you can say, the issues which are coming from diseases of viral origin or coming from feed origin, and then the contemporary changes what we are seeing in the genotype structure, and also you shared about the interventions or the approaches what we need to change, because the change is the only constant factor. And you reminded us that how important is to have a very robust diagnostic infrastructure to tailor-made the vaccination programs, and it needs to be very scientifically designed and judiciously designed. So, sir, that great words of wisdom you have shared. And sir, we have questions in our chat box as well. So maybe I can just read out for you, and you can share your feedback. So one question from Dr. Santosh is around the antigenicity changes. So it read like this: despite regular vaccination, there are major problems with vaccine failures. A change in antigenicity of infectious pathogen is one of the reasons for most of the vaccine. So what measures are being taken to address this particular issue? So maybe, sir, you can share from your experience on this topic, sir.
What I said, that we have to find out which type of virus is prevailing in your area, and find out. We need to carry out, maybe with the help of some universities or some institution like IVRI, trying to find out what type of sequence it is, whether it is homologous with the vaccine which we are using, or there has been some change in the virus. So because that only by sequencing only we can find out what changes have occurred in the field virus and why it is not responding to the existing vaccine. So either we need to add the virus into the field vaccine, or we have to update our vaccine as per existing virus challenge in the field.
Yeah. So, sir, this is very helpful, and I think there would be some questions. People must be, totally engrossed in your presentation and must be now contemplating your guidance and everything. So, while others may have any questions or clarification, I request our president and secretary and the EAC member, in case if they have any, feedback or viewpoint on the Dr. Mahajan's presentation. Yeah.
Good morning, sir. It's excellent presentation. Able to hear me?
Yeah, sure.
Sir, it is an excellent presentation covering all the diseases which are important for the industry. And there are one or two issues, sir. One is, for example, nowadays the rural poultry also increasing in population numbers. So, and they are having direct contact with the wild birds or migratory birds, and mostly they are not being vaccinated against many of the diseases. So what strategy we can adapt to vaccinate or you can say take care of such type of problems? That is one thing, sir. Another thing is, especially during outbreaks, even in the commercial farms, the dead bird disposal becomes a problem because of more number of birds. And in normal situations, whatever routine mortality is there, they're able to, handle. But when there is an outbreak, when birds are dying daily to 1%, 2%, in those situations, mostly the disposal is not that efficient, you can say, not enough. So to handle these things, what type of strategies we can adapt?
Yes, it's a very comprehensive question and good question. But the thing is that, when mortality is very high, definitely it's very difficult to dispose of the bird properly, because even if you have a small ditch, it will be filled up. So in Haryana, people mostly they provide the mortality to some fish farms, and which they use it as a mangur sort of fish, which they feed on these carcasses and utilize, let them off. But otherwise, now a lot of regulations are coming up from Pollution Control Board, and day after tomorrow we are having a meeting with Pollution Control Board chairman and secretary, and with all the farmers of Haryana, majority farmers of Haryana. They will tell their viewpoint how they are going to enforce these regulations, because that's going to be very tough for us poultry farmers now to throw away or to manage and how to avoid the pollution problems. So let us see what they say and what they answer they give to the farmers' queries, how they are going to have a very middle path, so that farmers are not affected. At the same time, there is no pollution to the greater extent to the environment. So both the issues are important. So so difficult. Well, we will need to find out. We need to find out solutions for that, because processing of dead carcasses for recycling of dead carcasses, I think it's not very common in India right now, because we may get it only during the mortality, but we may not get it regular basis. So somebody who wants to have a plant for processing, recycling, may not go. But in certain concentrated areas where a lot of farms are there, some people can think of some recycling plant, and here these they can be used by passing through the required temperature to destroy all the organisms, and then process it, not to destroy the protein source which is there, and can reutilize that protein source in recycling in some form, which can be used in pig feeding or some other animal feeding. These are some of the things, but we have to think of some solutions to this, all sitting together and find some areas where we can work together.
Yeah. So Dr. Mahajan sir, as I guess there are quite a number of questions coming. So the next one goes like this, again from Dr. Santosh. They want to understand that antimicrobial resistance is a major concern in veterinary and human medicine. The government is drafting antibiotic use standard. How will chicken farming cope up with this change? Your thoughts, sir?
Yeah, that is there, because we have seen, as I told you, that farmers, they are not aware, they do not know what antibiotics. They say antibiotics is the last resort to control the disease. They normally they are starting from the 4th generation straightway. They do not have idea that we have to start with the mildest one in the first week of life. They will start from levofloxacin in very first week. So we need to guide, we need to tell, and we have to educate ourselves also that these antibiotics, which are 4th generation, they should not be used. They should be used very sparingly when there is a severe outbreak only, not for routinely or not for prevention purposes. For prevention purposes, we should use only milder, like we should prefer probiotics in the first week of life. If there is some change, we can start with some neomycin or doxycycline combination. That is the mildest one. And then if it is not controlled, then only we can go for higher antibiotics. But it is always better to go for culture sensitivity and find out which antibiotics will be effective in your situation, and use the one which is easily available, which is cheaper, which is not going to cause much harm to the bird. And so that way we have to recycle. We keep on cycling the antibiotics. We have not to continue using one antibiotics batch after batch. So that is, we have to advise farmers on those aspects.
Yeah. Thank you, sir. Next question is from Professor Rachna Jain. She's saying that mutation is one of the major causes for the inability to perform in any vaccine. How this will be overcome for prevention of any disease?
Well, that is true, but you see, most like right now, till now, we were having Newcastle disease, which Lasota was proving very effective, and even kill vaccine was having mainly Lasota. So that was very effective and giving protection. But now, because of mutations, the new genotypes have emerged. So what we can do is, if new genotypes have emerged, what companies can do, who are already vaccine manufacturers, and with the help of government, they can tell the government that this is a new genotype which has emerged and this need to be added into the killed vaccine, inactivated vaccine, which we can do at the earliest. Otherwise, produce a new live vaccine, it's a long process. And again, live vaccines, they are difficult to produce for all strains. So what only thing is we can do is into the existing killed vaccine, we can add the virus, which is now prevalent in the area, new genotype has emerged, and ask the government. I think government will be coming forward to help you, to the companies, to add these type of viruses which are already prevalent. But provided government wants some reports from some good labs or institutions that this has been published in somewhere. Yes, sir.
Yeah. Thank you, sir. Next one is from Mr. S.S. Prasad. So their remark is like this: today we are observing variant strain of genotype 7 and 13 of RD virus in field conditions. At present, in India, genotype 7 and 13 official vaccine are not available. So how to control this problem, sir?
Well, I think now many, people, genotype 7 has been added into some vaccines. They have mentioned even on their labels. It is there, I think. But genotype, because genotype 7 was, it had come long, maybe 5, 6 years earlier. So it has been added into the vaccine. Some of the vaccine they are putting in the label, but many of the companies, they are not putting it, maybe may not be putting it on the label right now because they have to pass through the process of permission from Government of India. So they are in the process, and hopefully this will be done in coming time, within few months time. I hope this will be available to the poultry industry soon. All latest things, but Dr. Garware can put his light on this because he knows he is a man from vaccine company. So these type of things, they keep on updating those things. So hopefully people will be able to get the kill vaccine with all inclusive. All
Yeah, thank you, sir. And I know people might have further more questions, so they can keep on putting into chat box. Sir, 1 or 2 minutes, I'll just take to share a viewpoint on the AMR. So since I also work on the behalf of INFA, so a path-breaking initiative INFA has taken recently in compilation of the quantification of the antimicrobial usage in livestock sector. So we all say that there is an antimicrobial resistance, and however, when we talk about data specificity, that was the gap, sir, that how much is the antimicrobial agents are used in our country. So we and our team has done this task and quantified the data as per our own methodology, available resources. And we saw that in 2021 over 2020, so there has been a very minuscule, you can say, growth of the antimicrobial consumption in our country. Also, sir, when you compare this usage versus the biomass which is available in our country, so it's very, very minuscule, sir. And what has been contributing for this is, one, there is a lot of managemental interventions happening. For example, good managemental practices you mentioned, sir, feeding program, genetic improvement. Of course, the prevention is better than cure approach. A lot of, you can say, focus on doing the vaccination in time, that is contributing to reduction of the antimicrobial agents in poultry sector as well. In addition, sir, antibiotic usage is a cost to the farmer. So whatever interaction we have with the farming community, say they will try to do their best possible through interventions of biosecurity management and feeding program and vaccination program. Yes, sir. In situation of sickness of the animal, yes, antimicrobial agent are used, and I totally agree with you, sir. It has to be very judicious, prudent, and selective uses of antimicrobial agents, and one needs to be exercise a lot of caution, and also using the tools like ABST for selection of the antibiotics. So anybody on this call, if you wanted to learn more about that report, what we have compiled, which about the quantification of the antimicrobial agent, not only in poultry but also in the livestock sector, INFA can be reached out, and our therapeutic subcommittee has led this project, and we also have submitted this report to the Department of Animal Husbandry as well, sir. So this is just 2 minutes. I wanted to share this insight.
Yeah, that's very good initiative which INFA has taken. That because these type of studies will give them the impression that industry is working in that direction, and we are not enforcing, we are not the cause of AMR. We are helping government to minimize it. So that's good that INFA has done it, and they are otherwise they keep on blaming the industry without having any much of the proof.
Absolutely, sir. And in last year also we did a webinar talk on this specific topic, and all the experts suggested that data should be supported while making these claims. Like, and I'm also very much willing to learn any data point which are available for the quantification of the antimicrobial usage by species in our countries. So coming back to other questions, I was expecting that people have got a lot of excitement with your talk. So a couple of more questions. One is, kindly highlight the issue of backyard poultry vaccination raised by Dr. M.R. Reddy, sir.
Well, backyard vaccination is again definitely a very challenging one because they have small units, and we have to, maybe companies, vaccine manufacturing companies can help in that direction by making some smaller aliquots of vaccines, like 100 dose vials or something like that, or thermoresistant strains like we have in Israel and other countries, where you do not require help of a refrigerator to keep that vaccine, and you can use particularly Newcastle disease vaccine, because Newcastle is the one which is the most dangerous virus particularly for the desi birds or rural poultry. So that way, either we should have a thermostable virus, or we have to have a small vaccine and dosages dispensed from where they are getting the bird. They can be given directly from that place, like CPDO or government poultry institutions, where they are selling the birds to these small units of poultry to the farmers. So along with that, they have in the inputs, they should provide the vaccine and give them the training how to put it in intraocularly. That is important, because farmers need to be advised how to use it in intraocularly, because that will help.
Sure, sir. And sir, the next question is from Sir Muthu Kumar. Sir, the question is this: sir, evaluation of avian influenza vaccine, AI vaccine, how it is being done, and if so, where we can find lab for the evaluations?
Well, in India, there is no lab right now. Evaluation only at AAHP can do. Bhopal laboratory is authorized to carry out those. So, but in our meeting, we had discussed it long back. So there were a lot of questions, and government had said straight away that we are not going to deal anything related to avian influenza as such, H5N1, only H9. So they have agreed right now only on H9N2, not on H5N1. But as world over, I also emphasized in that meeting also that world over, almost all countries are having H5N1 vaccine stocks available in their countries to meet the catastrophe or challenges in incoming future time. Because even in US, you will find if there is any outbreak in any state, they have all the stocks available, and immediately they vaccinate around that state the vaccine. So that is for emergency purpose. At least we should have this type of mechanism, that we should have the vaccines available, so that if anything outbreak occurs in somewhere, we can use that vaccine after 3 square kilometer area or somewhere, whatever government decides about. But as such, there is no institution. Only high security animal lab can give some clues. Only government say that we are not touching on HPI, H5N1 vaccine right now. Only a low pathogen.
Yeah, sir, I'm aware that we are 2 minutes over than the scheduled agenda of Q&A, but since the program is meant for our participants, so I'm taking the liberty to have one more question. So the question is, any state government has policy to vaccinate poultry like ruminant vaccination?
Well, I think no, not for poultry, because you can say the poultry is not considered at all in any of the government sectors, except for backyard farm. Only backyard farm is there only. Rest is managing. Rest people are managing at their own. Government is not providing any help in that direction. No, just like government and national control program for FMD or brucellosis. No, it's not like that in poultry.
Thank you very much, sir. And I request all the participants, should you have any further questions, so please write to our event ID where Dr. M.R. Reddy's email is mentioned. So Dr. M.R. Reddy will collate any further questions and pass it on to Dr. Mahajan, and we'll arrange you the suitable replies accordingly. Thank you, Dr. Mahajan, sir, for such a exciting presentation, and the amount of questions you saw that clearly speaks about the interest and the curiosity you generated through your talk. So going ahead with the proceedings, I will request our Honorable President, Dr. Kataria, sir, to make a very exciting announcement, which all the AAHP members were being waiting for quite some time. So Dr. Kataria, sir, over to you.
Thank you very much, Dr. Makhija. Really, we had a very, very nice presentation by Dr. Mahajan. He discussed the real issues, and even the audience has put up some very pertinent questions. Where was the last question about the AI vaccine evaluation? What he said is correct. Government has permitted for H9, but not for H5. So the evaluation AI vaccine is maybe a H9, not H5. Government has not permitted to produce anybody H5 vaccine. So if it is H9, it should go through DADF to the NISAD laboratory for evaluation. And only those who are permitted to produce the vaccine, they will be sending, and their vaccine will be accepted. And other manufacturers, suppose even for any experimental or something, they are not supposed to produce it without biosecurity laboratory in their place. Now, Dr. Mahajan made a very nice presentation, and you know what he said about in the beginning, what we had ND very nicely controlled with the F and R2B, and followed by 1 or 2 sorts of inactivated vaccine, and ND was controlled. But now, listening to Dr. Mahajan, 18 to 19 genotypes have emerged out. But my concern is definitely they are coming out, and there is some escapes with the vaccine also. But even the present vaccine, they give good amount of cross protection, like one from audience has asked about the 7 and 13. If that is the case, they have 7 and 13, whether the vaccine which are prevalent have been tried to control these things. There is some variation, but there is a good protection, cross protection is also there. Am I correct, Dr. Mahajan?
Yes, there is cross protection, but not 100%.
Yeah, cross protection is there because there is no vaccine available for other serotypes, and even we don't have the epidemiology about other serotypes. How many are prevalent, which is prevalent, in which area it is prevalent. So to go for any vaccination, it will be a very time-taking and long process to go for this serotype vaccination, because not very many countries, of course, few countries only, they do vaccination with the ND, which are having endemic, particularly the Asiatic form. They go for vaccination, but most of the developed countries, they have eradicated even NDV, so they don't go for vaccination. So this we have to be very careful. Similarly, what he said, with the emergence of even chicken anemia, now it has also become endemic, and it goes unnoticed because we don't do that. I mean, thorough vaccine postmortem of the young chicks dying, whether we examine the bone marrow, we examine the thymus. So those early low mortality continued, and there will be damage. Of course, in chicken anemia, damage to the lymphoid system is, reversible to some extent, but immunosuppression is there, and then secondary complication comes, and the process of wasteful investment goes on. So that is how the chicken anemia, I remember it was permitted once to use inactivated vaccine produced by some of the leading vaccine manufacturers. but I don't know what is the present situation, whether that is being used or imported. And what Dr. Mahajan said about the vaccines which are being used prevalent, what I said in the introductory remark, that is one of the cause of emergence of mutation. So until unless we do the epidemiology and genotyping, just adapting to the vaccine and importing the vaccine, definitely we are going to get some monster in the days to come, which will be simply uncontrollable. So we have to be very judicious, whether that is the industry or that is the government organization, because poultry is very limited in the hand of government organization, DPR and CRI. We have a limited stock. Industry is having, you said, in lakhs they keep. So they have to be, very cautious. And whenever there is a disease, they should get its genotype by sending it. They may not be having facility, or they may not be having the competency. But those laboratories which are doing the genotyping, they should get it, those things, genotype, and identify which virus is prevalent, which pathogen is prevalent, which vaccine will be available, will be effective. And similar is the case of LPI and HPI. LPI has become endemic. HPI is coming frequently. So definitely one assortment is a very good multiplication process in this AI viruses. Something new will come up and we will not be having any answer to it. So we have to be very religious in reporting of the disease, which DADF, even they have made a system to report the disease, but one has to report. If industry has to progress, they have to report about the disease, what are the prevalent, what are the diseases coming out, what are their genotype, which vaccine is effective, which vaccine is not working well. Simply, if something is not working, we cannot blame the vaccine. We have to see the environment. Vaccine is effective in the clean environment. If environment is not clean, it is already suppressed with the mycotoxin. Earlier, we don't give that much weightage to the mycotoxin. But nowadays, mycotoxin has become very important. With so many immunosuppressive agents, immunosuppression with the mycotoxin and then superseding with the other pathogens, leading to the present catastrophe in the poultry industry. It is a continuous loss. So what are the things what Dr. Mahajan mentioned about the monitoring of the feed, monitoring of the mycotoxin is very important, and it should be done at the feed processing plant itself. Then the feed should go out. Perhaps these are very important things. We have to monitor the strain, we have to see the vaccine. For everything, if you go for vaccine, that is not the answer. Vaccine is definitely a protection. But we have to follow other things like biosecurity, prevention is the basic and fundamental things to set up any poultry farm, which we adopt. Nothing, definitely more than 50% of the problem is solved. And of course, the source of any chicks, anything should be from renowned, established sources. With this, I think it is very clear we have to follow religiously the poultry husbandry. Pollution will come, how to dispose of dead birds. That is very important. We have not only think of our interest, we should have a system in place for disposal of carcasses within the farm premises, how to dispose it of, not to contaminate even our water resources. That is very important. Not to go and throw and dump in some area from where the vultures, the crow, they will take it away and spread the infection. Perhaps these are very important points which Dr. Mahajan has emphasized, which every farmer's industry has to adopt. Thank you very much. Thank you, Dr. Makiza.
And Kataria sir, thanks for your great words, sir. We also wanted to hear the announcement from you, sir.
Yeah. Now, after this, we are coming to our business of AAHP. Due to Corona, we were not able to conduct our physical conferences. Now we are planning to have it in the month of February next year, and maybe second or third, fourth week somewhere, clubbing with the one of the weekends and Friday, Saturday, like that. Dates and theme will be communicated very shortly, and I am happy that Dr. Chatterjee is with us. He has accepted our request and invitation to host the conference. So it will be in Hyderabad only. We are trying to have it in Hyderabad, and Dr. Chatterjee, as director of DPR, has kindly agreed to host it. And I request Dr. Chatterjee now to make a few words about this conference, hosting of conference.
Thank you, sir. My esteemed colleague, Dr. J.M. Kataria, Director, former Director, CARI, Dr. N.K. Mahajanji, former Dean, Lala Lajpat Rai University, Veterinary University and Animal Science, and today's esteemed speaker, Dr. Makija, esteemed colleague from industry, Dr. Sanjay Gapkare, probably he is also present in this meeting, in this conference. Other esteemed colleagues from the industry, my colleagues from my own institute, Dr. M. Reddy, and other participants. It was really very excellent presentation from Dr. N.K. Mahajanji. He has covered all the aspect of disease aspect of the poultry sector, and thereafter comments from Dr. J.M. Kataria and Dr. Makija. So, of course, I have very little knowledge on health because I don't belong to health aspect. But some of the things, of course, we are doing in our institute that I will tell. But if you see the poultry industry, poultry sector in the world, China is producing around 586 billion eggs, and India is producing 129 billion eggs. That means 22% of the China's production India is producing, and around 7% of the total egg production of the world India is producing at present. And I do not know whether now at present India has surpassed America or not in terms of egg production. Probably it is either close to America or probably it has surpassed America. Either in the second or third position. Meat production, we all know we are in the fifth position. We are producing around 4.6 million tons of poultry meat, which is, if we see the total meat production, including all the species, all the livestock species, poultry is producing more than 50% of the total meat production of the country, that is 9.29 million ton. And the growth in poultry industry, all of you know that we have mentioned in different conferences, in different forums, that egg production in our country that has been 67 times as compared to 1950 egg production. 67 times. This is the only agricultural commodity where this increase has been made. And probably poultry is the only sector, that is the first sector that has taken a shape of industry among all the agricultural commodities. So thereafter, the poultry egg production recently that has been termed as silver revolution. So probably all of us we know, and that is a great thing. And my sincere thanks, compliments, and congratulations to all the farmers and all the people who are engaged directly or indirectly in the poultry industry or the poultry sector as a whole. So poultry is growing maybe 6 to 7% in layer, 8 to 10% in broiler. But in spite of the growth, there are certain challenges we are facing. Number one is disease. That was the topic of today. Number two is feed, escalating feed cost. Number three is AMR, Dr. Makija was mentioning, and Dr. Kataria Sahab was also mentioning. And number four is poultry cage welfare. So the welfare aspect that has also been taken care of earlier by BIS, where I was the chairman, and then by DADF. And thereafter, the verdict from the Delhi High Court that has come, so that has been almost solved. The cage welfare aspect that is solved. Now, AMR aspect, of course, Dr. Makija has mentioned about the INFA activity, whatever they are doing, and ICR and the university, Agricultural University, Veterinary University, they are also having a network project that I wanted to mention here, that is INFA. So in this INFA project, that is a network project supported by ICR and under the this by FAO, where most of the Animal Science Institute as well as some of the Fisheries Institute, they are the collaborator, they are the participating institute for seeing the impact of the AMR, antimicrobial resistance, from different species of livestock, from different species of fish, from soil, and so on and so forth. So probably in the near future, ICAR also will say that this is the impact from different sectors of animal science and fisheries sector on the contribution of AMR. Then, of course, the most important disease that has been covered in detail and excellent presentation by Dr. Mahajan, and that was covered, of course, thereafter the comments from Dr. J.M. Kataria ji. So what we are doing in our institute, and there are some questions, very beautiful questions by the participants, maybe from my own colleague, Dr. M.R. Reddy, maybe from Dr. S.S. Prasad, maybe from Muthu Kumar, maybe from Dr. Archana, whatever name I can remember so far. They have put many good questions. So I'm not going in those details, but the thing is that we know that viral genome is very small genome, and mutation in the virus, that is a problem. That is causing a problem in the vaccination failure. That may be in infectious bronchitis, that may be in ND, that may be many other diseases. Say infectious bronchitis, earlier there was some vaccine, but now infectious bronchitis nephropathogenic strain that has come. And then MD also, we are working in collaboration with some international avian health company. So probably some other variants through mutation that has come. So in collaboration with the industry, our institute, Directorate of Poultry Research, they are also contributing. Of course, we are not producing, we are not developing any vaccine, but we are helping the health industry to say that these are the strains for which vaccine is needed. So that is one aspect. Then AMR already INFAR, we are working in different species, not only in poultry, in other species also we are covering as per the INFAR mandate. And the last thing that Dr. J.M. Kataria already has mentioned, this institute, Directorate of Poultry Research, is going to host the conference, next conference of AAHP in 23rd and 24th February of next year in Hyderabad. So organizing secretary will be, of course, Dr. M.R. Reddy, maybe one or two more colleagues from this institute. So I welcome and all my colleagues presenting presented in this meeting. So please come and join, and we'll discuss in detail about different aspects of poultry, whatever challenges are there, whatever these things are there. So in detail, everything we will discuss. So AHP will be hosted in next year, 23rd, 24th February by DPR at Hyderabad. Thank you so much.
Thank you, Dr. Chatterjee.
Thank you, Dr. Chatterjee, sir, for your words of enlightenment and encouragement to entire AHP community. And we are indeed privileged and honored to have this event happening at your institute. And this institute has been very friendly to all the research and development happening in the poultry sector. And we will be really looking forward to have this event under the leadership of you and all the collaboration of Dr. M.R. Reddy sir, his team. So with this, we move to the last topic of the this program, that is a very important aspect of thanking the entire AHP participant of this webinar and the community. I request our EC member, Dr. R. Saravanan ji, to please propose a vote of thanks.
Yeah, very good afternoon to you all. I am very proud privileged for proposing the vote of thanks for this particular webinar. First of all, sincerely, I would like to convey my sincere thanks to the President, Association of Avian Health Professional, AAHP, Dr. J.M. Kataria, sir, who is the motivating force for moving forward of this particular association. And I also propose my sincere thanks to Dr. Sanjay Gavkre, Vice President, AAHP, and also the guest speaker, Dr. N.K. Mahajan, who is former Dean of Luas Hisar. For sincerely, we convey our thanks to the guest speaker for sharing his more than 3 decades of experiences, and definitely it will be very helpful for the industrial people, academician, researcher, each and every participants of this particular webinar. And I also would like to convey my sincere thanks to Dr. Mahija, who excellently conducted this program. And I also thanks Dr. R.N. Chatterjee, Director DPR, for providing the logistics to conduct this program. And I also convey my sincere thanks to Director Dr. R.N. Chatterjee, sir, for kindly accepting our AHP conference as the venue for the next conference. And I also sincerely thank Dr. M.R. Reddy, sir, who is instrumental for arranging each and every program of AHP, and he is the backbone of conducting each and every program. I also sincerely thanks the other EC members of AHP. And last but not least, I would like to sincerely thank the participants. Currently, there are 42 numbers of participants, and most of the webinar was attended by more than 80 people. Exactly 83 members were participated in this particular webinar from various academics, students, researchers, and also the industrial people. So, lastly, I would like to convey my thanks to each and everyone who helped directly or indirectly for the successful conducting of this particular webinar. Thank you.
Thank you, Doctor, for expression of vote of thanks. So with this, ladies and gentlemen, we complete our webinar on time. So thank you so much for all your support and encouragement to AAHP. We look forward to meet you in next 2 months again with